Okay, it's my pleasure to introduce the next speaker. The next speaker is Joern Alexander Quent. He's from Fudan University, Shanghai. He'll be speaking about precise mapping of the neural landscape underlying recognition memory.
Yeah, thank you. First of all, I'd like to thank you for coming here. Obviously, the organizer for organizing this event. And I'm very excited to share our latest work on this topic. So for those people who might not know what recognition memory is, let me give you a very quick explanation. So imagine tomorrow you come into this place again, you see this guy, and you have the thought, hey, I've seen this person before. Well, that's basically you're just experiencing recognition memory. In the laboratory, we have various ways to study this and other types of memory. For instance, we can use free recall. We can use confidence ratings from the participants. There's something called the remember no procedure, or still probably the most common way of studying recognition memory is just a simple yes or no recognition task, which is also the one that we use in this experiment. The way to study recognition memory in the brain, the most canonical way is still what is called the old new, or the retrieval success effect. And for that, it's basically important to understand kind of like the lingo that comes from the signal detection framework, where if a participant calls a new stimulus new, we call that correct rejection. If someone calls an old stimulus old, we call that a hit. And for the retrieval success effect, you just subtract one from the other, essentially. Also, what's important for the rest of the talk, for those who don't know, is in the signal detection framework, you have a signal and a noise distribution. D prime is kind of a measure of accuracy and the separation of these distribution. And participant can also vary their criterion and bias their response to be more strict or more liberal, essentially. And generally, we believe that recognition is a mixture of recollection and familiarity, where, for instance, recollection would be something, in the example earlier, if you think, well, I know this person because his talk was so boring, you would engage in recollection. While if you just have a general feeling of having seen this person before, that would basically be familiarity. And we believe, like research has shown that like recollection might be more represented in default mode regions of the brain, while familiarity might engage more frontoparietal areas. So what do we know? What kind of networks are supporting recognition memory? One of the most prominent one is certainly the parietal memory network that have been identified in task and resting state work. We know from very early work that if you vary the ratio of old and new stimulus, we still find a retrieval success effect in these regions. We also find it if participants only are rewarded for recognizing new images, so making those the targets. And we also find it if we only require responses for old or for new or for both. So it's a very robust effect that doesn't seem to suggest that it's pure targetness that is driving these retrieval success effects. However, there's also work that shows in like an attentional task, like a postnat task, if you have an invalid cue, you find the same kind of regions that also show more salient, high activity, more salient effect. Relatedly, in participants that show a stricter response bias, we also find stronger retrieval success effects. And the whole network, and even like the slightly more extended, is also activated in a really simple target detection task that have very minimal memory requirements, which does suggest there might be something that goes beyond pure memory retrieval, and that's what we're trying to find out. So our question basically is, what is the influence and saliency in driving the retrieval success effect? We used a continuous recognition task where we present each image 3 times. It's a very large data set. We collected data from 20 people. I'm only talking about 18 here at the moment, where it's 5 sessions, each with 12 runs. So in total per participant, we have 3,840 trials and 96,000 on total. Throughout the experiment, you see we have very high response rate, pretty good signal-to-noise ratio measures, and acceptable movement parameters. And one thing, as a consequence that we repeat each image 3 times, we have a lot of new images at the beginning and very few images that are new at the end of the experiment. But most importantly, the accuracy over time is relatively stable. Though there is a great variability across participants, but they continue to be engaged across the task, essentially. The image processing, it's 70 fMRI. We use ACP minimal preprocessing pipeline, multimodal surface matching, and single trial beta estimates with no additional smoothing. So the 2 millimeter is what comes out of ACP pipeline. And all of that is basically done to precisely map and localize recognition memory at the group, but also an individual level. And when we move to an individual level, we get exactly the kind of regions that we would like to find. So it's like precuneus, middle cingulate cortex, that is exactly kind of matching the results that you would get in like meta-analysis and like hundreds of studies that have been done of this kind of work before. And it basically frontal parietal regions that show retrieval success effect, and the reverse would be more like secondary visual regions. However, if you appreciate the short video, if we look at individual subjects, they're highly variable. And what our data allows us to do is, like sometimes that could be just a problem of like signal to noise ratio at this individual level, but we have enough trials to look at things at the individual level. So that's what we've done here. So like on each side of the contrast, we have up to 6% of the whole brain being significantly after stringent FDR correction at the individual level. And this is not a function of whether the participant was good or bad. So the 2 worst participants and the 2 best participants show relatively similar cluster size. But the first take-home message here is already, we find no part in the brain that is significantly activated across all people when we look at the overlap of these clusters. And when we try to quantify how participants, how similar brains are across participants and to the average, we find correlation values that range from almost like high to almost non-existent. And to show that that's actually a meaningful individual-specific neuroanatomy, the participants that show a less strict response criterion, they correlate better with the average brain when you exclude themselves, obviously. So that's why we now look at the more individual level, and what we wanted to find what's the role of saliency. We have like one way to look at it, what we call mnemonic surprise. So for each trial, we basically calculate how surprising is this trial type. So for instance, at the beginning, most old trials are surprising. At the end, most new trials are surprising. That's basically what is illustrated here and like shown in this equation. And when we look at this in the by using the individual retrieval success maps as our clusters, we find that at each participant, the same clusters show higher activity when something is more surprising. And like, yeah, we find the reverse for like the kind of like more novelty preferring clusters. Interestingly, another way how we think something can be more salient in our experiment is if something has been presented more recently. So for each old stimulus, we basically calculate in minutes the time between each presentation, so like between the second or the third, for instance. And that can vary in the order of minutes, like it can be presented in the same run, but it also can be, the difference can also be on the order of days, essentially. And here we find a very similar pattern, a bit more noisy, but here now with all these orange clusters, we basically find that high activity if a stimulus has been presented very recently, which really interestingly also matches results from kind of a slightly different type of memory, which is autobiographical memory, which shows exactly the same, exactly the same type of regions that are active, essentially. And yeah, with that, I like to end, basically. So the take-home messages is one, the retrieval success rate is like highly robust on the group level, but we also find a really high level of heterogeneous neuroanatomy, and that neuroanatomy I think is very meaningful, and we lose this if we average across subjects, which creates these kind of maps. And we do find evidence that on the individual level, if we really use these retrieval success clusters, we find that they are sensitive to salience when we measure it through like mnemonic surprise, but also temporal recency. And with that, I'd like to end by thanking you for your attention, obviously, the wonderful lab that I'm part of that makes all of this work possible. And yeah, if you want to complain about anything or you just want to have a chat, you can come to my poster, which is later on as well.
Great. I think we have some time for some questions. And I don't think any have come through on the app yet. So maybe I'll ask the first question. I was wondering if you looked at if there's any differences in gyral or sulcal patterns in in your individual anatomy. You've shown very convincingly that the functional activation patterns vary greatly across individuals. I was just wondering if there's any underlying anatomical differences that you can relate those functional differences to.
I mean, the short answer is no. But the main motivation of, for instance, using multimodal surface measure, I haven't really explained that well, is it using like more sources of information to really try to align individual the brains across individuals better, like using resting state and like myelin maps, etc. So we try to already eliminate as well as we can kind of like the differences that might already exist in like other types of like analyses. But yeah, I'll probably look at that. That's a very good suggestion.
Okay, maybe I'll ask another question, unless other people want to come to the mic. Have you looked at any lateral parietal regions, like angular gyrus?
No, I mean, like, my main interest for this work was kind of like the posterior midline regions, but like we do find kind of very similar things, I think, like in angular gyrus as well, to be honest, like in the lateral parietal regions. But we really were interested in the posterior midline regions because like there's been kind of like recent kind of increased interest in that, so we wanted to kind of what are these things actually doing basically at the moment.
Great. And then maybe one really quick question. The question is about the relationship between pattern separation and high fidelity memory and how it's linked to linked to recollection. Maybe that's not a quick question, but maybe you can give a couple of your thoughts very quickly.
I mean, like, so one thing that I definitely, that might be tangentially related to that question is you could obviously wonder, like, both in terms of, especially for the temporal recency, that, or what if I'm just looking, like, if it's something that is recent, that is basically a very fresh memory, well, the further away, it just becomes more similar to a new memory, like a new image again, so basically people just forget. And we do find that within the posterior midline regions, the overall patterns match pretty well, but in other, like for instance, secondary visual regions, it doesn't really. So I'm not entirely sure that for instance, would explain everything, but that's the best I can do for this question. Thank you so much. Thank you.